All right, so now I'd like to talk about a third imaging mode, and that is lightning deconvolution. So this is an add-on to our sort of most basic imaging mode, which is to take an XY plane through your sample and then move the focus, take another XY plane, and so on. So we have a stack of slices in the Z direction, and we can use that to make a 3D reconstruction of the whole sample. So that's basically what confocal is for. But it turns out there's a bit more information as you move from slice to slice, as you go through the depth of the sample. There's also information there on where the photon is really coming from. In any light microscopy system, photons will tend to spread out, and it can be hard to see exactly where they were coming from if you just look at a single slice. So here we have software that essentially replaces the signal back to its source as you move through a Z-stack. And so that's called deconvolution. And Leica has their own implementation of this. It's called adaptive deconvolution, or lightning would be the brand name for that. So I personally had not done a lot of deconvolution before, and I was always a little bit suspicious about any type of post-processing of images that can tend to introduce artifacts. So I was a bit suspicious, but now I'm really completely sold on lightning. We've been using it quite a bit, and it's very effective. Now, it's not for everything, and I'm happy to discuss where it would be a good application for you and where it would not help you very much. It does take a bit of time, but it's it's not live, it's almost live. So you collect your Z-stack, you wait about less than a minute, and then the deconvolved lightning version of that Z-stack will appear, and then you can process that over. It usually takes about 5 or 10 minutes to process it and export it. So it's not instantaneous, it's a lot more rapid than deconvolution has been in the past. So what you see here is the raw image and then the deconvolved image after the lightning processing. And you can see it, it's quite a different image in a number of ways. And in the most obvious way, it appears clearer, there's less noise, and the features appear sharper. All right, so we're going to take a little deeper dive on this. I'm going to show you what this sample is here, and then we'll talk a little bit more about it. So this sample is a fly egg chamber. They're from fly ovaries that I stained in order to test our ability to look at 5 different dyes at once. And these are the 5 different dyes or markers that are present in the sample. And what we're going to do is we'll look at all 5 as we just saw, but we're going to focus on this GFP marker for early endosomes, and then this UV marked stain for filamentous actin. And we'll just look at the pair of those in the later slides. All right, so if we go ahead and take a slice through this epithelium that surrounds the developing oocyte, that's what we see here. So we're looking down individual cells and their nuclei here. But if we take a cross section, as is shown here, this type of plane, we can take a cross section through the epithelium. Now, to do this, we have to focus rather deep into the sample. And the problem with confocal is that the deeper you go, the more the light has to be scattered before it hits the objective. And so your sample gets dimmer, the appearance of your sample gets dimmer and dimmer and dimmer as you go deeper in. So standard confocal, like what we have here, is only best for rather shallow distances. So that's just to point out that this is a rather more difficult imaging situation than you would encounter, for example, if you just had a cell culture that was flat on a cover slip. And here we're very much zoomed in on just a few cells here in this epithelium, again in cross section. So this is my best approximation for, or my best example for the comparison of standard imaging and lightning. So I've taken an optimized standard image, and then at the same point, I ran the lightning process. So this is in contrast to what I showed you before. This is not the pre and post lightning. This is the standard imaging and then the post lightning. So the standard image you can see is perfectly good. You can see individual features there, these endosomes, membranes, microvilli, actin filaments. And then after, so that would be the final step of standard imaging. Here, doing the lightning process where you're collecting things in a slightly different way and then doing the deconvolution, you can see, I think pretty clearly here, that a lot of the haze is gone, and that haze is both noise and out of focus signal. And then you see that the structures are very much tightened up. So you can see a lot of resolution of individual actin bundles here, and individual microvilli are a lot clearer. The membrane itself, the membrane marker is very much thinner than you see here. So you get a lot of resolution, and it removes a lot of noise as part of the deconvolution process. Now, looking at this, I was still not entirely convinced. I do kind of like the original unprocessed data. And so I'm going to show you the experiment that kind of converted me so that I would really strongly recommend lightning under certain circumstances that I'll show you here. So in this is the same sample, and now we're taking a slice through here. It's similar to this one, slice through here where you're glancing through the apical side of this epithelium where there is a lot of early endosomes, and then we've also stained for actin. So now we'll just look at these two dyes. So we have early endosomes in magenta and actin in green. These are the microvilli. But if you get a plane underneath the microvilli where you're seeing just early endosomes, what you might notice is that there are little dots of actin associated with the early endosomes. So this is the full-sized image. We can get a lot of information with the lightning image. And here what I'm going to do is now zoom in on a few of these early endosomes. Okay, so this is the same type of image, and just zooming in. So now we're looking at the pre-lightning and post-lightning deconvolution. So again, this is the raw data for lightning, and then this is after it's processed. And so what I want you to focus on here is not just the losing this background haze that we talked about, but what actually happens to these objects. So again, the raw data for this object is shown here, and then after deconvolution. So this endosome, what you'll notice is that the size of the object doesn't change. If you compare these two, size didn't change. The boundaries got sharpened up a bit. And it's kind of nice. You can see that this is a vesicle, and now it has a lumen, which would be very hard to distinguish here. So it's giving us a lot more information about this, but the size didn't change from the raw. And now let's look at the actin dots that are associated with these vesicles. And you see in each case, the actin dot is roughly half the width of the endosome that it's associated with, somewhere on the order of about half the size. But after deconvolution, so for example, if this was just taking this in Photoshop and sharpening it up, that would not change the size of the objects. But after deconvolution, what you can see is the size, as I mentioned, for each endosome does not change, but the size of the actin dots does change, and it gets much, much more compact. Look at this one here. Here the signal is very much spread out. Here the signal from wherever it has been spread out in 3 dimensions now through the Z direction, it's been calculated back and replaced back to its source. And so the actin dots got very much more compact. So it really is treating the objects differently based on their the pattern of signal as you move through the Z stack. So the question then is it inventing something that's not there, or is this actually getting you closer to reality? So with any type of post-processing, you want to be hopefully moving towards reality from what you've collected here. And so there's no inherent way to know from this image, but luckily there was a previously published paper that can give us a little bit insight into this that I came across. And so these structures, little actin dots on endosomes, this has been characterized before. And so this is a paper out of HeLa cells. And so if we assume this is a pretty conserved process, which it seems to be, we can use the sizing information from this paper. So very helpfully for us, they had performed transmission EM, and so they could get the exact sizes of the vesicles and the actin dots. There's little actin dots there by EM. And the bottom line here is that the sizes that they found in HeLa cells matches exactly with what Lightning gives us. And so it turns out that the actin patches are at about 50 to 100 nanometers, and that's what we're seeing here. There's a micron scale bar. And so indeed we do seem to be going back more towards the reality of what these things really look like. Now what's nice about this is that the typical resolution limit for light microscopy without any processing or any sort of optical tricks would be over 200 nanometers. So it would be hard to resolve things that are less than 200 nanometers. They would tend to blur together. But here we're clearly seeing as distinct items that are on the range of 100 to 200 nanometers versus 50 to 100 nanometers. Again, it's easier to resolve things if they're in different colors, but you can clearly see that these are very sharply resolved by this system. And so we're clearly getting well below the 200 nanometer resolution limit, which is why these are structures are blurring out. We're not seeing them as 100 nanometers with standard imaging. All right, so what I've just taken you through is spectral fingerprinting, FLIM, and deconvolution. And so I think that these are three methods that really can work together or separately to help to mine more information out of your samples and get a little bit more data above and beyond standard confocal. And so now I want to just finish up by saying a little bit about zoom. And so let me jump forward here. All right, so I would say right now we're in October of 2020. And so we're right in the middle probably of the pandemic. And I would say that about a year ago, before the pandemic, I would have never thought that at this point we would be using any type of conferencing software on the confocal. With our old system, at least, that would have been a big no-no. So we did not want to have the old system even on the network at all. Now, the current system is safe to be on the network. However, you typically want to avoid having lots of other software going at the same time as you're trying to do all of this type of, things like FLIM processing and lightning processing. You don't want to have the computer trying to do other things, multitask while you're doing those. So in my mind, I always said we're not going to have any other types of software running while we're on the confocal. But the pandemic has kind of forced us to do things we wouldn't otherwise do, right? So we wanted to do some training. I have a confocal training class here. And I wanted to do that in a safe way, so try to do it remotely. So we tried out Zoom on the confocal, and I was really kind of blown away by the results. So we can just simply launch Zoom while the system is running, and we can zoom out from the system to anywhere, to the whole class. And then we can record those sessions. And this is a screenshot of the Zoom recording. And you can see the resolution is such that you can see everything that you need to see. The software, the Leica software, there's so much there that the font is very small, but the Zoom recordings basically are good enough to see all the information. All right, so this lets us now do two things that we wouldn't do before. First of all, we can be on the scope, do a session, and then anybody, anywhere can watch what's happening on the screen. And most of the action on the confocal is happening on the screen. All right, so the remote user can't see what's happening over on the physical microscope itself, unless you have a laptop there that's showing what's going on. But you can do that as well. All right, so this means that if you want to be a user from a different institution, you could, for example, send a student. Student could be on the scope running it, and I could be helping them. And then as a PI, you could be sitting at home and monitoring exactly what's going on. And then you could then provide feedback. Your student would have a laptop present there. And so we could communicate over the laptop, and you could be watching exactly what we're seeing on the screen. So I think that will really increase the ability of outside groups to use the system effectively. So in other words, everybody doesn't have to come to the scope and do things. And that means that we could even collaborate. I could even get a sample by FedEx and pop it on there, and you could basically tell me what to do with it. This would also be very good for outreach. So you're sitting on the scope, doing all of these different processes, lightning, FLIM, and so on. And that could be broadcast out to your lab group or to a course, a lab course, or to any type of STEM outreach that you might be doing, maybe a high school group or any other type of group that's interested in seeing what science is actually all about. The scope is so visual and so dynamic that I think it provides a really nice example of experimentation. You can basically do an experiment just by moving a slider up and down. All right, so if anybody has any of those types of applications, outreach and things like that, I would be more than happy to talk that through with you. And again, you can then also have, if you send a student to use the scope, they can archive exactly what they've done on your Zoom, and then you can check back and see exactly what happened. And that can be valuable because the scope, the options are so complex, it can be hard, even if you're taking good notes, to know exactly what steps you took with a given sample. All right, so those are the main points that I wanted to get across today. And so if you think that you might want to be a user, please go ahead and contact me. If you want to just have a consult and see if it will work for you, we can set that up over Zoom. And thanks for listening.